So the last talk will be given by Jishnu.
Jishnu, yes.
Jishnu.
Good afternoon, everyone. I'm Jishnu. I'm an assistant professor at the Center for Systems Immunology at Pittsburgh. So I'm a computational systems immunologist. I run a computational systems immunology lab where we think about two things: how you can integrate high-dimensional multi-omic data sets in the context of the immune system using predictive and interpretable machine learning, and how you can also use biological networks to integrate different kind of omic data sets to uncover molecular phenotypes of immunological disorders. And we do this across disease contexts in infectious, autoimmune, and alloimmune diseases. But what I'll be talking about today is some of our interpretable machine learning work that we did recently. And I mean, this was really, really interesting. This is an absolutely fantastic conference. But one theme that has kind of emerged is we have all of these deep learning methods, and they focus very, very deeply on prediction, but not necessarily inference. So what are we actually learning? So this, what I'm going to present today, is on the other end, a statistical machine learning approach, which prioritizes inference. We still do very well on the prediction front, but what are we actually learning in the context of explainable AI or interpretable machine learning? And in human systems immunology, when I wear my statistics hat, I say, okay, prediction and inference are both important, but when I wear my biology hat, we really need inference beyond prediction. So this is something that we published in Nature Methods earlier this year. This is following up on a larger body of work with essential regression and other papers, but I'll be focusing on our Nature Methods paper and then show you some downstream applications. We call this slide Significant Latent Factor Interaction Discovery and Exploration. And this was done in conjunction with key collaborators, Alok, Bob, and Amanda, and the work was co-led by two awesome graduate students in my group, Javad and Hanshi. So what is the slide model? So this is a latent factor regression model where rather than moving from the x's, the features, to the y, the response, I mean, all of you are familiar with regression, we first move from the features to the latent factors, the z's, and that's an unsupervised step. And then we move from the latent factors to the y. Now, the biological intuition is very, very simple. Biology doesn't care about what we can measure. In 2024, we have all of these different data modalities: single-cell RNA, single-cell attack, proteomic, metabolomic, but biology doesn't care about what we can measure. Clearly, the true answer lies in something that is hidden, hence latent factors. Now, this is not some great insight. Latent factors have been around in the statistical literature for 2 decades or more, but there were some very nice, interesting properties of what we did with latent factors that allowed us to do this regression model. So step one is we set up a latent factor discovery framework with identifiability guarantees in parameter space without making any assumptions regarding data generating mechanisms, without making any assumptions regarding orthogonality of the latent factors. So we solve this. So other methods, let's say non-negative matrix factorization methods, are non-identifiable. On the other hand, if you have methods like principal components regression, obviously the principal components are orthogonal. So we can prove, this was work done in conjunction with theoretical statisticians at Cornell, we can prove unique identifiability of the latent factors by allowing them to be overlapping without making assumptions regarding data generating mechanisms. The way we do this is we solve this as a constrained optimization problem where we anchor each latent factor based on what we call our two pure variables, two variables that belong only to that latent factor and no other latent factor. So of thousands of features, let's say you have 100 latent factors, so 200 would be, so each factor would have 2 anchors. Everything else can be shared or not shared in a data-dependent fashion. This is done using the underlying covariance matrix. Now that we have identifiable latent factors, we regress to outcome using these. So here regression makes sense. In an NMF setting where the latent factors themselves are moving around, regression does not make as much sense. But these are stable latent factors, so we can regress to outcome using these. And here what we did, we can prove guarantees on the regression coefficients. But remember that the latent factors themselves can be overlapping. That reflects true biology. So they can be multicollinear. So rather than using the p-values on the individual regression coefficients, we use an overall method for false discovery rate control developed by Emmanuel Candace and colleagues called knockoffs. This is obviously very, very widely used in a number of different contexts. So we control overall false discovery rate. The key adaptation was knockoffs was originally in the observed space. We adapted to the latent space, to the z's. So we identify what we call significant standalone latent factors. Now, using hierarchical OR logic, we test nonlinear up to pairwise interaction terms and apply knockoffs again. So we identify significant standalone latent factors and then significant interacting latent factors. And we have a model where we come down to a necessary and sufficient set of latent factors to understand the response or infer the response. Note that I have never used the word causality so far, and I'll come to that. As a statistician, and we've actually coupled this with causal graphical modeling, you have guarantees even in terms of causality. But as a biologist, I would never claim causality without showing it in a perturbation system. So I say inference or putative causal. So SLIDE has many unique aspects, including identifiability, no assumptions regarding data generating mechanisms. It's not restricted to any single cell or bulk data modality, can handle nonlinearity, correlated latent factors, FDR control, inference beyond prediction, and I'm like comparing it to other methods in this space, including variational autoencoders, partial least squares, principal components, lasso, and so on. So now let's actually look at what we did with this method. So the first data set that we started off with, this was in conjunction with Bob Lafiatis' lab, who's a local scleroderma expert. We had single-cell RNA-seq data from skin biopsies, and we asked what are the significant latent factors underlying disease severity. So here, disease severity is scored by punch biopsies on the skin, where the skin is scored on a scale of 0 to 3. 0 means completely normal, 3 is the thickest. So it's a surrogate measure for the disease severity called the modified Rodnan skin score. And we said, not only can we predict, but what can we actually infer or learn about underlying disease severity from the single-cell RNA-seq data? And of course, the first thing we looked at with k-fold cross-validation and permutation testing, how well do we predict? This is kind of the necessary, but to me, the less interesting piece. So SLIDE and essential regression, the two methods that we developed, predict this as accurately or better than state-of-the-art methods. But what exactly are we learning? What is the interpretable part? So this is the first example that I'm showing of the kinds of latent factors that we get. So this is a busy slide. I'll walk you through it. So each box that you see is a latent factor. It's a context-specific group of genes. This structure is inferred using the underlying sample covariance matrix. This is not a predetermined gene set or pathway. We do not have any biological priors. We are simply doing this in a data-dependent fashion. So now, within each latent factor, you have these genes. The color represents the cell type of origin. So you have keratinocytes, fibroblasts, T cells, and so on. Left of the dotted line, you have genes associated with higher disease severity, right of the dotted line with lower disease severity. The green boxes are the significant standalone latent factors. The purple ones are the interaction terms. So without using any biological priors, we were able to recover, for example, in the top right, the pro-fibrotic fibroblast differentiation. This is a special subset of SFRP2 fibroblasts or myofibroblasts known to be involved in scleroderma disease pathogenesis with CCL19, IGF-BP5, well-known markers. We also see crosstalk between myeloid inflammatory signaling and interferon signaling like thrombospondin, CCL19 again, ISGs. So these were well known. Without using any biological priors, we were able to recover. This is what's well established in the field. But we converged on two very interesting things. The first was the crosstalk, not just of fibroblasts and myeloids, which in the field is believed to be the true driver of disease pathogenesis, but also a role for keratinocytes. So we see these interaction terms with keratinocytes. And that was very, very intriguing. And that is something we actually validated in downstream. I'll show you one slide on that. That's a new mechanism involving crosstalk of fibroblasts, myeloid cells, and keratinocytes that we validated. The second was the T cell responses, which I won't talk about. So SLIDE latent factors we rationalize are meaningful with robust effect sizes. Here I'm showing you like the empirical null. That's the blue if you just pick a size match number of latent factors. And then the green is if you fix the standalone but you shuffle the interactors, the model is at the tail end of that. So you can calculate tail probability, a p-value. So we then said, well, how good are we overall? So we are giving you specific vignettes, but let's say we see how many known genes do we recover. We do better than MOFA plus or VAEs, and in terms of effect sizes of each of these latent factors, we again do better than these. And the latent factors are meaningful. This is one example of where we validated the keratinocyte signature using protein staining. So in the scleroderma, you have these patients with higher levels of keratinocytes. Now we said, let's push the bar a little harder. So are we actually getting something mechanistic? So this is a human perturbation experiment. So a subset of these patients were treated with tofacitinib. That's a pan-JAK inhibitor. So we had patients who were responders and non-responders, and we said, can we infer latent factors that discriminate responders and non-responders? And without using any prior knowledge, the key latent factor that the model found was this IL-6 STAT signaling-centric mechanism, reflecting that depending on how well you respond to the pan-JAK inhibition, you respond or do not respond. So this is recapitulating a known mechanism of action. This is another piece of evidence showing that we are actually capturing something meaningful. Now we switched systems. That was a organismal disease phenotype. So now we went to a NOD model, this is in conjunction with Alok's lab, of type 1 diabetes, where we looked at a cellular phenotype. We said, well, we have paired single-cell RNA-seq data and TCR-seq data, where each cell was labeled based on the extent of clonal expansion of these CD4 T cells, so unexpanded single, low expansion, or medium to high expansion. And we said, can we now understand this cellular phenotype in a disease context? And again, we can predict very well. So the top left plot is in a k-fold cross-validation setting. Slide and ER are predicting clonal expansion very, very accurately, but that's not really the main piece. The main piece is what we learn, what is interpretable. So we see three key latent factors, one involving markers of naive CD4 T cells, some well-known markers of naive like L-selecting or CCR7, celebrated activation markers, inhibitory receptors like LAG3, PD1. But we see these context-specific group structures emerge. And why are they important? They are capturing higher effect sizes than any univariate canonical marker genes. So this is kind of a thought experiment where we said, well, if I talk to my immunology colleagues, they will come up with LAG3, TIGIT, PD1, CD200. These are well-known inhibitory receptors, markers of activation. They are all somewhat higher in the expanded relative to the unexpanded. L-selectin, CCR7 are higher in the unexpanded, but it's not a slam dunk. On the other hand, if we look at these latent factors, they clearly stratify based on extent of clonal expansion. So we are really capturing robust, explainable effect sizes using these context-specific groups. Now, I made the claim across biological domains. That's a big claim. So have we really done this across biological domains? So we've been talking about single-cell data modalities. Now we switch data modalities completely. So this is in the context of schistosomiasis infection, where we have this platform technology called antibodyomic profiling, where using a Luminex-like assay, we can quantify the response of a polyclonal pool of antigen-specific antibodies. So for different antigens, we are measuring the isotype subclass profiles binding to Fc receptors, and we are saying, using this interpretable model, what exactly do we understand of the latent factors, humoral latent factors driving schistosomiasis infection? This was done in conjunction with Anirudh's lab, co-led by Trirupa in my lab and Anushka in Anirudh's lab. This is now in press, should be online in a couple of weeks. So I'll keep it very brief just to show you high-level vignettes. The first is people know in the context of helminth infection that you have higher IgG4. So if you do the standard DE kind of analysis, the only thing that you see is IgG4 and some Fc receptor binding. But when we ran the slide model, we see a very, very significant model. So I'm showing you the model performance with k-fold cross-validation and permutation testing, but it's not just IgG4 and FCR binding. We also see a crosstalk between IgG1, Fc binding, as well as an IgA1 dominant latent factor. So we are finding novel mechanisms or inferring novel mechanisms. But how real is the inference? And we wanted to put it to test. So we said the first thing is let's validate this in an orthogonal cohort. So we discovered these latent factors using a cohort in Brazil. We tried to validate the signature using a completely orthogonal cohort, Kenyan cohort, and the model still does well. I mean, it goes from 0.9 to 0.7s, but the key thing is both the structure of the latent factors. We discovered the unsupervised structure discovery as well as the prioritization were only done using the Brazilian cohort. We did not see the Kenyan cohort at all, and we still have signal. So we are capturing something that is extrapolable across contexts. What we also were able to do is we were able to look at different mechanisms of active, passive, and prior infection, and I won't go into details of that, but all I'll say is it's not just IgG4 in classical helminth infection. We find a key role of IgG1, and also the unbiased profiling tells us that CD63 and calumenin are the two major antigens that people focus on, but actually find this SM25 specific responses are a critical discriminator, not of active and prior passive, but within prior and passive, and that can help early detection. So this is really, really important. Again, switching gears, this is also in press now. We've worked with Amanda Poholik's lab to identify spatial micro-niche specific cellular programs in a murine model of allergic asthma. I'm only showing you a couple of slides. There's a lot of data in this. I'm focusing only on the spatial data. This is 10x Visium as well as slide-seq data. So these are mice exposed to intranasal house dust mite. That's the allergen. They continuously inhale the allergen, and they have basically a severe asthma phenotype. They are profiled at day 3 and day 5. So we asked this question, which a lot of people in the spatial field are asking: what are these spatial environments or micro-niche specific cellular programs? And what we find is at day 3 and day 5, spatial regions enriched for Th2 and TF8 cells, so these are primary cells, this is in a lymph node, so you have B cells at the boundary, T cells at the center, including TFH and Th2 cells, have divergent cellular programs. At day 3, for the TF8 cells, interactions with the B cells are critical to maintain the TF8 cells. This is known biology. But the intriguing part was we found this in this latent factor Z13. The actual pure variable, and it's a key driver, was IL-2RA. And the IL-2RA, IL-2 pathway, we hypothesized this was not known, is critical for Th2 differentiation. At day 5, the TFH B cell signature still stays the same, but the Th2 cells are now engaged in migration to the lung. And with murine models, it was actually validated that spatial niches of IL-2 synergize with IL-10 to drive lung migratory Th2 cells in asthma. So again, this is the kind of mechanism that we are inferring. And for the last piece, I'll talk about characterizing dynamic gene regulatory networks in primary human B cells. So we are a part of the IGVF consortium, so it's a multi-PI grant that Harinder and I have. So this was work co-led by multiple people in our labs, where we are looking at B cells, so naive B cells. This is an in vitro system, but with primary cells, they are differentiated into activated GC-like extrafollicular-like cells. And we said, can we now identify cellular programs and gene regulatory network circuits that are discriminating these? So the slide model learns de novo cellular programs discriminating granular cell states. Here I'm showing you the GC versus PB model using really one standalone significant latent factor and one interaction. We can almost completely separate these. This is very nice properties. We tried this like learn from a male donor, predict for a completely orthogonal female donor, learn across dataset modalities from single-cell RNA alone to multiome. It all works well. In a lot of cases, DE analyses completely fail for this. But what is more biologically interesting is slide captures combinatorial control of transcription factors in state-specific GRNs. So we can superimpose these context-specific cellular programs that we learn with inferred gene regulatory networks to identify combinatorial control of transcription factors. I'm just showing you one example in the PB state, how BLIMP1, actually PRDM1, works with multiple different transcription factors to control the PB state. This is unpublished work, so I'll not talk about it anymore, also in the interest of time. So in summary, SLIDE can integrate high-dimensional multi-omic data sets with no assumptions regarding data-generating mechanisms to identify significant latent factors underlying immunological outcomes. These are putative causal, and some of the inferred mechanisms have been validated using perturbation systems or model organisms, comes with rigorous statistical guarantees regarding identifiability and corresponding inference, FDR control, and the discovery of nonlinear hierarchical relationships. Using SLIDE, we have identified causal factors of a range of phenotypes from organismal to cellular, molecular, across contexts in health and disease. I talked about scleroderma, type 1 diabetes, asthma, schistosomiasis. We also have other work in myocardial infarction and COPD that I didn't talk about. And finally, I would like to thank my lab. None of the work that I do would be possible without my lab, and of course, my funding sources. And thank you very much for the invitation to present here. I'm happy to take questions.
Any questions?
Thank you. Very nice talk. Could you please elaborate a little bit more, like about the idea how you create these latent factors? You know, why 2 anchors, not 3 or 4? What's the biological idea behind this?
Great. So I'll answer it in 2 parts. The first is we create the latent factors based on essentially the underlying covariance matrix, and we are clustering into overlapping clusters the underlying covariance matrix. Getting to Y2, that was the least stringent assumption. So we basically said we don't want to make any assumptions. We cannot prove identifiability without making any assumptions if we allow them to be orthogonal. So 2 was the very least. So you could absolutely have 3 or 4 or more, but 2 is the least that we need to prove identifiability guarantees of the latent factors. And biologically, what we are saying is each of these context-specific groups, cellular programs, humoral profiles, is anchored by a couple of things that belong only to that latent factor and no other latent factor. So we are allowing, let's say, to use a concrete example, let's say you have a certain cell type, let's say in CD8 T cells, you're looking at 4,000 or 5,000 genes, you get 100 latent factors. You're only assuming that 200, so for each of the 100 latent factors, for each factor you have 2 genes. So 200 are locked into each individual, all the other 4,800. So more than 90 to 95% of your data in practical examples can be pleiotropic, reflecting the true underlying biology.
Okay. Thank you. Let's thank the speaker.
Thank you.